Welcome back to the deep dive. We are jumping right into the fascinating world of microbial control today. We're going to unpack the really intricate regulatory blueprint that bacteria use to manage their entire genetic library.
Yeah, it's incredibly complex stuff.
And we're starting the story, interestingly, not in a lab book, but with a real-world application. A life-saving bacterial light show.
That's right. It's a fantastic example of how elegant these systems can be.
Right.
Researchers, they designed an E. coli biosensor. Basically, they took a promoter sequence, one that specifically responds to the explosive compound TNT,
Okay.
and they fused it to the gene for green fluorescent protein, GFP.
Right, GFP. So if this engineered bacteria is nearby, say, an old minefield and maybe some TNT vapors leak out.
Exactly. The promoter recognizes that signal. It flicks a regulatory switch. The E. coli starts making GFP.
And the whole colony just lights up green under UV light. Wow.
It's genetic control being used for, well, rapid real-time detection. Pretty amazing.
It really is. But it also kind of brings us to our main mission today, right? Because while that TNT sensor is pretty much like a simple switch, on or off, your sources make it really clear that regulation in nature, it's rarely that simple.
Absolutely not. It's much more like a molecular rheostat, a dimmer switch. It's about finely tuning the amount of gene product needed, and the big reason, to conserve massive amounts of energy.
Okay, let's unpack this dimmer switch idea then. We're looking at how bacteria regulate the flow of genetic information at three main levels? Transcription, translation, and post-translation.
That's the framework. And yeah, how they use these mechanisms to stop wasting energy and materials.
Right. It's like the ultimate microbial efficiency plan, you could say.
Exactly. I mean, why build a whole complex protein pathway if the thing you need is already, floating around? Why bother?
Makes sense. So let's start at the most fundamental level. Transcriptional regulation right at the beginning, at initiation. We need to clarify what's being controlled. You have these constitutive genes.
Yeah, the housekeeping genes, they're just expressed continuously, always on, things the cell always needs.
And then you have the regulated genes.
Right, and these are only switched on when the cell actually senses a change, when it needs them.
Sure.
It saves a ton of resources.
Okay. And within those regulated genes, we can sort of categorize them based on their job.
We can. You often talk about inducible enzymes. These are typically involved in catabolism, breaking things down. They get induced, turned on by the presence of their substrate. Think lactose breakdown.
Okay, inducible breakdown. And the other type?
Repressible enzymes. These are usually biosynthetic in that they build things up like amino acids.
Yeah.
They get repressed or turned off when their final product is already available.
So if there's plenty of tryptophan around, the cell stops making more.
Precisely. Why waste the effort?
Now, the actual decision makers here, the ones doing the switching, are these DNA-binding proteins, right? And they often work as pairs, as dimers.
Yes, often dimers. And they bind to very specific DNA sequences called palindromes. These are inverted repeats, like reading the same thing forwards and backwards on opposite strands.
Why that specific structure, the palindrome?
Well, it has to do with how the protein dimer physically interacts with the DNA double helix. They need to fit just right. They often use a structure called the helix-turn-helix motif.
Okay, describe that.
It's basically two short alpha helices connected by a sharp beta turn. One of those helices fits snugly into the major groove of the DNA that allows the protein to read the specific base sequence there.
Like a key fitting into a lock.
Exactly. And what determines if the key turns, if the protein binds or lets go, is something called allosteric control.
Allosteric control.
This means a small molecule, an effector, could be an inducer, could be a co-repressor, binds to the protein somewhere else, not at the DNA binding site.
Okay, away from the business end.
Right. But that binding causes the whole protein to change shape, a complete conformational shift. And that change dictates whether the protein can still grad onto the DNA or if it have to let go.
And that shape shift is the core of whether we get negative control or positive control.
You got it. In negative control, the regulatory protein is called a repressor. When it binds to its site on the DNA, called the operator, it physically blocks RNA polymerase from starting transcription. It's a roadblock.
Okay, negative means blocking.
And in positive control, the protein is an activator. It binds to a different site, usually upstream of the promoter, called the activator binding site. And instead of blocking, it actually helps RNA polymerase bind. It promotes transcription.
So it gives polymerase a helping hand, essentially.
Precisely. It often facilitates a binding, making transcription more likely or happen faster.
Let's look at some classic examples. The lac operon, that's the textbook case for negative control, right? Specifically, negative inducible control.
That's the one. The lac operon has the genes needed to break down lactose, lac-C, lac-C, lac-A.
And normally, if there's no lactose around,
If there's no lactose, the lac repressor protein is active. It binds very tightly to the operator sites. There are actually three, O1, O2, and O3. This binding even loops the DNA.
wow, bends the DNA.
Yeah, it physically bends it, creating a steric hindrance that blocks RNA polymerase. So no transcription. The genes are off.
But then lactose shows up.
When lactose enters the cell, a small amount is converted into allolactose. That's the actual inducer molecule.
Ah, not lactose itself.
Right. Allolactose binds to the lac repressor, and that binding triggers the allosteric shape change we talked about.
The repressor changes shape.
And it can no longer bind to the operator DNA. It falls off. The roadblock is gone.
And RNA polymerase can now transcribe the lac genes. The cell starts making the enzymes to use the lactose.
Exactly. It's induced by the presence of its substrate.
Okay, then we flip the logic for the TREP operon, the model for negative repressible control. The cell's asking, why make tryptophan if we already have it?
Yeah. Exactly. Total waste of energy. So here, the TRIP repressor protein is synthesized in an inactive form. We call this the aporepressor. It can't bind the operator on its own.
So it needs something else.
It needs the corepressor, which in this case is tryptophan itself. When tryptophan levels in the cell get high,
It acts as the signal.
right, tryptophan binds to the aporepressor. This binding activates the repressor complex. Now, this active repressor can bind to the TREP operator,
And block transcription.
blocks transcription of the tryptophan synthesis genes. The pathway shuts down because the end product is already there. It's repressed by its product.
Okay, that covers regulation at the start, but then things get, well, even more complex. You mentioned stopping transcription after it's already started.
Right.
That seems counterintuitive. We're talking about attenuation.
Attenuation, yeah.
Yeah.
It's brilliant, really. And it relies on something unique to bacteria. The fact that transcription and translation happen at the same time and place. They're coupled.
Right, no nucleus separating them.
So the ribosome can jump onto the mRNA while it's still being transcribed by the RNA polymerase. Attenuation uses this coupling. The cell starts making the mRNA, but it uses the speed of that very first ribosome as a way to check local resource levels almost immediately.
How does it do that? In the TREP operon specifically.
It happens in the leader sequence of the mRNA called trPL. This leader region comes before the actual structural genes. It contains a short coding sequence for a leader peptide, and critically, this peptide has two tryptophan codons right next to each other.
Two codons. Okay, that seems important.
Very. The leader RNA also has 4 regions, 1, 2, 3, and 4, that can fold and pair up in different ways, forming hairpin loops.
So what happens if tryptophan is scarce?
If tryptophan levels are low, the ribosome translating that leader peptide stalls when it hits those 2 treptocones because there aren't enough charged tRNAs carrying tryptophan.
It pauses, waiting for tryptophan.
Right. And crucially, this stalled ribosome physically covers region 1 of the leader RNA. This prevents region 1 from pairing with region 2. So instead, region 2 pairs up with region 3.
And 2.3 pairing forms?
What? It forms an antiterminator loop. This structure doesn't stop RNA polymerase. It's a signal to keep going. So transcription of the whole operon continues. The cell needs to make tryptophan, so it keeps the factory running.
Okay, that makes sense, but what if tryptophan is abundant?
If tryptophan levels are high, the ribosome doesn't stall at the tripcodons. It zips right through them and continues until it hits the stop codon in the leader peptide sequence.
Oh, it moves faster.
Much faster. And where it stops covers region 2. Now, because region 2 is blocked, it can't pair with region 3. This leaves region 3 free to pair with region 4.
And 3.4 pairing forms.
That forms a terminator loop. This structure is a signal recognized by RNA polymerase. It's an intrinsic, factor-independent termination signal.
So it tells polymerase to stop. Right there.
Immediately, polymerase falls off the DNA template. Transcription terminates prematurely, before the structural genes are even made. The cell already has tryptophan, so it stops the process early.
Wow, so it's like a second layer of control on top of the repressor?
Exactly. And when you combine the trap repressor with attenuation, the cell can decrease transcription by like 600-fold. It's incredibly fine-tuned control. Real molecular genius.
It really is. Now, beyond proteins controlling things, you also mentioned riboswitches. These aren't proteins at all.
Cool. Riboswitches are sensory regions within the mRNA itself, usually in the 5-foot untranslated leader region. They act like direct sensors. They can fold into complex shapes, and part of that shape can bind directly to a small effector molecule.
So the RNA itself is the sensor and the switch.
Precisely. Take the riboflavin operon in Bacillus subtilis. The leader mRNA has a region called the RFN box. If levels of FMN, which is derived from riboflavin, are low, the mRNA folds into a shape with an antiterminator loop. Transcription continues.
Makes sense. Need more riboflavin.
But if FMN levels are high, FMN itself binds directly to that RFN box in the mRNA. This binding forces the RNA to refold into a different shape, one that includes a terminator loop.
And that stops transcription, just like an attenuation.
Just like it. Halts transcription early. No protein repressor involved, just the mRNA and the small molecule effector. It's remarkably efficient.
That's controlled during transcription. What about later at the translation stage? Do riboswitches work there too?
They do, especially common in gram-negative bacteria. These translational riboswitches work on a similar principle. Effector binding causes the mRNA leader to refold.
But instead of forming a terminator loop,
Instead of affecting transcription, the refolding physically hides or occludes the Shine-Dalgarno sequence.
the ribosome binding site.
Exactly. If the Shine-Dalgarno sequence is buried within the folded RNA structure, the ribosome simply can't bind. Translation initiation is blocked. The RNA is made, but no protein comes from it.
Another way to shut things down quickly. Okay, and then there are also small RNAs, or siRNAs.
Right. siRNAs, sometimes called non-coding RNAs or ncRNAs. These are relatively short RNA molecules, maybe 25 to 500 nucleotides long, and their main job usually is to regulate the translation of specific target mRNAs. Mostly, they inhibit it.
How do they do that?
They typically work by base pairing with the target mRNA, often near the ribosome binding site. This binding can either block ribosome access directly, or sometimes it recruits enzymes that degrade the mRNA.
So they interfere with the message. Are there different kinds?
Yeah, broadly, you can think of cis-encoded siRNAs, which are transcribed from the DNA strand opposite their target gene. They're basically perfect antisense complements. And then trans-encoded siRNAs, which are encoded somewhere else in the genome.
And those need help finding their target.
Often, yes. Trans-encoded siRNAs usually have only limited complementarity to their targets, so they often rely on a protein chaperone called HFQ. HFQ helps stabilize the siRNA and facilitates its pairing with the target mRNA.
Can you give an example of siRNA action?
Sure, a classic one is the OMPA-If-MIGHEF system in E. coli. It relates to porins, the channel proteins in the outer membrane. E. coli needs OMPA-F, which makes a large pore when nutrients are scarce, like in dilute environments.
Okay, big pore for scavenging.
But in high osmolarity conditions, like the human gut, a large pore is risky. It lets harmful things in. So the cell prefers to make OMPC a smaller, safer pore.
Makes sense. So how does the siRNA fit in?
When osmolarity gets high, the cell ramps up production of an siRNA called MICF. MICF, often helped by HFQ, binds specifically to the OMP mRNA.
The mRNA for the large pore.
Right. This binding blocks the translation of OMPF protein. So high osmolarity leads to MICF production, which shuts down the synthesis of the large OMPF pore, allowing the cell to prioritize making the smaller, safer OMPC pore instead.
That's really elegant coordination. It shows how different layers like osmolarity sensing and siRNA regulation come together.
Absolutely. And, just briefly, before we scale up to global networks, we should remember post-translational modification, even after a protein is made, is not necessarily active or stable.
Right. The story isn't over yet. The cell can still tweak the protein itself.
Exactly. Things like adding or removing a phosphate group, phosphorylation or dephosphorylation, can act like an instant on-off switch for enzyme activity. Or the cell can just decide to destroy the protein altogether using proteases. It's another level of fine-tuning.
Okay, so we've seen control at initiation, during elongation, at translation, and even after translation. Now let's zoom out. How does a cell coordinate responses when, say, a single change requires adjusting dozens or hundreds of genes all at once?
That's where global regulatory systems come in. These are systems that control many genes or operons simultaneously. The collection of genes or operons controlled by a single global regulator is called a regulon.
A regulon, okay. And a common architecture for these global systems is the two-component signal transduction system, or TCS.
Yes, TCSs are incredibly common in bacteria. They're the primary way bacteria sense and respond to their environment. They have two main parts, as the name suggests.
The two components being?
First, a sensor kinase. This protein usually spans the cell membrane. It has an external part that detects a specific environmental signal, like a nutrient, a toxin, or osmotic pressure. When it detects the signal, it performs an autophosphorylation. It adds a phosphate group to one of its own histidine residues.
Okay, sensor kinase detects and tags itself with phosphate. What's component 2?
Component 2 is the response regulator. This protein is typically in the cytoplasm. The sensor kinase quickly transfers that phosphate group from its histidine to an aspartic acid residue on the response regulator.
So the phosphate gets passed along.
Right. And getting phosphorylated usually activates the response regulator. Once activated, it can then do its job, which is often binding to specific DNA sites to either turn genes on or off, controlling the regulon. Sometimes it might interact with other enzymes instead.
We actually just saw an example, didn't we? The NVZ-on-OMPR system controlling the porins.
Exactly. NVZ is the sensor kinase detecting osmolarity. OMPR is the response regulator. When NVZ phosphorylates OMPR in high osmolarity, OMPR turns on the OMPC gene, small pore, and, using help from mic-ssRNA, effectively turns off the OMPU, large pore. Perfect example of a TCS integrating multiple signals and outputs. And while TCS is common, sometimes you see more complex versions called phosphorylase systems, involving multiple proteins in the phosphate transfer chain.
Okay. Another way to control big sets of genes involves the core machinery of transcription itself, the RNA polymerase, using alternate sigma factors.
Yeah, this is a really powerful global control mechanism. Remember, the core RNA polymerase enzyme can't recognize promoter sequences on its own. It needs a sigma factor subunit to guide it to the right starting spots.
Right. Sigma finds the promoter.
Well, bacteria usually have several different types of sigma factors. The main one, the housekeeping sigma factor in E. coli, is sigma. It recognizes the promoters for most essential genes.
But there are others.
Oh, yes. For example, sigma recognizes the promoters of heat shock genes. So if the cell experiences sunny heat stress, it rapidly increases the amount or activity of sigma.
And the polymerase, now loaded with sigma, ignores the normal promoters and just transcribes the heat shock genes needed for survival.
Precisely. It allows the cell to instantly redirect its entire transcriptional program towards dealing with that specific stress. There's sigma-della for flagella and chemotaxis genes, various ECF, extracytoplasmic function, sigmas for sensing stress outside the cell. It's like changing the lens on the polymerase to focus on different sets of genes.
A very direct way to manage large regulons. Now, tying a lot of these global responses together are second messengers, small molecules made inside the cell.
Exactly. Small molecules produced internally in response to some external signal or internal state. They act as intracellular signals that can trigger widespread changes.
Let's talk about the classic example, catabolite repression in E. coli. This involves CAM-MP.
Right. This explains that phenomenon called dioxic growth. If you give E. coli both glucose and lactose, it will always use the glucose first. Completely.
It ignores the lactose.
Ignores it. Then, after the glucose is gone, there's a short pause, a lag phase, and then it switches on the lac operon and starts using lactose. Glucose is just the preferred, most efficient carbon source.
So how does the cell know the glucose is gone and it's time to switch? This involves the second messenger CAM-MP, right, and CAP?
Correct. The key is the glucose transport system itself, the phosphotransferase system, PTS. When glucose is abundant and being transported, a component of the PTS called enzyme IIA is mostly unphosphorylated. In this state, it actually inhibits the enzyme adenyl cyclase, so CMP levels are kept low.
Okay, high glucose, low CAMP.
But when glucose runs out, enzyme IIA becomes phosphorylated, IIPA. This phosphorylated form no longer inhibits adenyl cyclase. In fact, it stimulates it.
So low glucose means adenyl cyclase gets turned on.
And it starts cranking out high levels of the second messenger, CMP. Now, CMP binds to another protein, the catabolite activator protein, or CAP. This CAMP-cap complex is the active form. It's the glucose is gone signal. This active complex then binds to specific sites near the promoters of many operons involved in using alternative sugars, like the lac operon, the AraOperon, and others.
And binding there helps RNA polymerase. It's positive control.
Exactly. Active CAMP-cap binding significantly enhances RNA polymerase's ability to bind and transcribe these catabolite operons. So for the lac operon to be fully maximally expressed, you need two conditions met.
What are there?
One, lactose must be present so the lac repressor falls off the operator, negative control lifted. And two, glucose must be absent, so CAMP-PK levels are high, CAP is active, and it binds to promote transcription, positive control applied.
It's like a genetic A&D gate. You need signal 1 A&D, signal 2.
Precisely. It ensures the cell only invests in using lactose when it's both available and the preferred glucose isn't around.
Very clever. Okay, another major stress response involving a second messenger is the stringent response. This sounds serious.
It is. Think of it as the cell's emergency brake. triggered primarily by nutritional stress, especially amino acid starvation.
What happens when the cell runs out of amino acids?
Well, during translation, if the ribosome encounters a codon for an amino acid that's scarce, the corresponding uncharged tRNA, one without its amino acid attached, will enter the A site of the ribosome.
The empty tRNA slotting in is the trigger.
That's the trigger. This event activates an enzyme called RALA, which is associated with the ribosome. Activated RALA then synthesizes special signaling molecules, the alarmones, ppGP-granazine tetraphosphate, and pentaphosphate.
Alarmones. Okay. And what do they do?
They have a massive global inhibitory effect.
Mm-hmm.
TPGPP basically signals times are tough, conserve everything. It directly interacts with RNA polymerase and dramatically decreases the transcription of genes needed for growth, especially those for RNA and tRNA synthesis.
Slows down ribosome production.
Exactly. It also inhibits DNA replication, translation, and other metabolic processes. The whole cell slows down, conserving resources until conditions improve. It's a critical survival strategy.
A global shutdown order. And there's one more second messenger system focused on lifestyle choice, c-di-GMP.
Cyclic di-GMP, yeah. This one is fascinating because it largely controls the decision between being motile, swimming around freely, and being sessile, sticking to a surface, often as part of a biofilm.
So swim or stick?
Pretty much. High intracellular levels of c-di-GMP generally promote the sessile biofilm lifestyle. It encourages the production of adhesins, sticky molecules, and extracellular matrix components, while often inhibiting flagellar synthesis and motility.
And low levels mean swimming.
Low levels favor motility. The levels are tightly controlled by two types of enzymes, deguanylate cyclases, DGCs, which synthesize c-di-GMP, and phosphodiesterases, PDEs, which break it down. These often have conserved domains, GGDEF for DGCs and EL for PDEs.
Can you give an example of how c-di-GMP works?
Sure. In Pseudomonas aeruginosa, a common biofilm former, high c-di-GMP activates certain transcription factors for biofilm genes. It also binds directly to a protein complex involving LAPD. This binding sequesters a protease called LAPG.
It traps the protease.
Right. LAPG's job is to cleave an important surface adhesin called CDRA. So when c-di-GMP is high, LAPG is trapped, CDRA stays intact, and the cell remains stuck in the biofilm.
But if the cell wants to leave?
If conditions change and c-di-GMP levels drop, LAPD releases LAPG. The freed LAPG protease then cleaves CDRA, cutting the anchor and allowing the cell to detach and swim away. It's the sessile to motile transition, controlled by this second messenger.
Incredible control over lifestyle. Okay, now let's look at processes where many of these mechanisms, TCS, second messengers, modification, all come together for complex behaviors. Chemotaxis in E. coli seems like a prime example.
Absolutely. Chemotaxis is how E. coli navigates towards attractants like nutrients and away from repellents. It's not directed swimming, it's a biased random walk.
Biased random walk.
Yeah, the cell alternates between smooth, straight-line swimming, called a run, caused by counterclockwise, CCW, flagellar rotation, and brief, random tumbles that reorient it, caused by clockwise, CW rotation. In an attractant gradient, the runs that happen to be going up the gradient are simply extended, while runs going the wrong way are kept short. This biases movement towards the source.
Okay, so how does it control the switching between run and tumble?
It uses a spiralized two-component system involving CHE, the sensor kinase, and CHE-I, the response regulator. These proteins cluster at the cell poles along with receptor proteins called MCPs, methyl-accepting chemotaxis proteins, which detect the attractants or repellents, and a coupling protein, CHE.
So these clusters are the sensing arrays.
Right. In the absence of an attractant, CHE autophosphorylates and rapidly transfers the phosphate to CHE, phosphorylated CHE, by the way, then diffuses to the flagellar motor and causes it to switch to CW rotation, inducing a tumble.
Tumbling is the default if nothing good is sensed.
Essentially, yes. But when an attractant binds to an MCP receptor,
The signal changes.
binding inhibits CHE's autophosphorylation activity. Less CHEAP means less CHEAP. Unphosphorylated CHE doesn't interact with the motor, so the motor continues its default CCW rotation, resulting in a smooth run.
So more attractant means longer runs.
Longer runs towards the attractant. But the really amazing part is adaptation. The cell needs to respond to changes in concentration, not just the absolute level.
It needs memory.
Kinda, yeah. It uses methylation of the MCP receptors. Enzymes share a methyltransferase, and CHE, a methylesterase, which is also a response regulator activated by CHEAP, modify the MCPs.
How does methylation help?
Higher methylation levels make the MCPs less sensitive to attractants and more sensitive to repellents. So even if the cell is in a high concentration of attractant, Share keeps adding methyl groups. This resets the signaling system, allowing the cell to sense even higher concentrations. It adapts to the current level and looks for increases. Che removes methyl groups when signaling is high, helping reset sensitivity the other way.
It's constantly adjusting its baseline. That's incredible.
It allows the cell to navigate gradients effectively over a huge range of concentrations.
Moving from individual movement to group behavior. Quorum sensing. Bacteria talking to each other.
Essentially, yes. Communicating using chemical signals called autoinducers to monitor their population density. When the density reaches a certain threshold or quorum, they coordinate gene expression, often triggering group behaviors like virulence factor production, biofilm formation, or famously bioluminescence.
Like in Vibrio fishery, that's the classic simple system.
Right. V fishery uses an AHL acetylhomoserine lactone autoinducer. Each cell produces a small amount, and it diffuses out. At low cell density, it just dissipates.
But in a crowd,
At high density, like inside the light organ of its squid host, the AHL accumulates. It reaches a high enough concentration to diffuse back into the cells. Inside, it binds to the transcriptional regulator LuxR.
and LuxR gets activated.
Activated LuxR then turns on the genes for light production, the Lux operon. It also turns on Luxi, the gene for the AHL synthase, creating a positive feedback loop that rapidly boosts the signal and light production once the quorum is reached.
Simple and effective. But some systems are way more complex, like in Vibrio Harvio.
Oh yeah. VRV is a great example of sophisticated quorum sensing. It uses a complex phosphorylase system, not just a simple TCS, and it responds to at least 3 different autoinducers simultaneously.
3. Why so many?
Well, one, HAI1, seems to be species-specific. Another, AI2, is produced by many different bacterial species, allowing for interspecies communication. And the third, CAI1, seems specific to the Vibrio genus. So it's getting information about itself, its close relatives, and the general bacterial community.
That's multi-layered communication. How does a phosphorylase work?
It's intricate. At low cell density, in the absence of autoinducers, the sensor kinases associated with these signals phosphorylate a cascade of proteins, LuxU then LuxO. Phosphorylated LuxO activates the production of small regulatory RNAs.
sRNAs again.
Yep. These sRNAs block the translation of the master quorum sensing regulator, LuxR, a different LuxR than in V fishery. So at low density, no LuxR, no light.
Okay, but at high density?
At high density, the autoinducers bind to their respective sensor kinases. This binding flips their activity. They stop being kinases and start acting as phosphatases.
They remove phosphates instead of adding them.
Exactly. They drain the phosphate out of the luxophosphorylate cascade. Unphosphorylated LuxO is inactive, so the sRNAs are no longer made. Without the sRNAs blocking it, the LuxR mRNA can now be translated.
LuxR protein is made.
And LuxR protein activates light production genes. Interestingly, in V. harvii, high-density quorum sensing also represses things like the type 3 secretion system, T3SS, the injection needle bacteria use to inject toxins. So it suggests a switch from individual attack mode to a more communal, perhaps biofilm-associated lifestyle at high density.
Wow, complex signaling integration. Finally, let's touch on how bacteria defend themselves, particularly against viruses, bacteriophages. This involves regulation too, right?
Absolutely. 2 key systems here. The first is like an innate defense. Restriction-modification, RM systems.
How does that work?
Bacteria produce methylase enzymes that add methyl groups to specific DNA sequences within their own genome. This methylation acts like a self-tag. They also produce restriction endonucleases, RE enzymes, that recognize those same specific DNA sequences, but will cut the DNA unless it's methylated.
So their own DNA is safe because it's methylated.
Right. But when a virus injects its DNA, which is usually unmethylated at those specific sites...
the restriction enzyme cuts it up, destroying the viral genome.
Exactly. It's a simple, effective way to distinguish self from non-self DNA and destroy invaders. Innate immunity.
But then there's CRISPR-Cas, which is more like adaptive immunity. It involves memory.
That's the amazing part. CRISPR-Cas provides bacteria with an adaptive, heritable defense system based on past encounters. It works in stages. First is adaptation.
What happens then?
If a cell survives a viral infection, specialized Cas proteins, Cas1 and Cas2, cut out small pieces of the invading viral DNA or RNA. These pieces, called protospacers, are then incorporated as new spacers into a special region of the bacterial genome called the CRISPR array. This array consists of alternating repeat sequences, and these unique spacer sequences derive from past invaders.
So the CRISPR array is like a genetic most wanted gallery of past viruses.
Kind of, yeah. It's a molecular memory bank. The next stage is expression and interference. The entire CRISPR array is transcribed into one long pre-CAR-RNA molecule.
Okay.
This pre-CAR-RNA is then processed by other Cas proteins into mature CAR-RNAs, CRISPR RNAs.
Sweet.
Each mature CAR-RNA contains one spacer sequence, the viral memory, and part of the repeat sequence.
So each CAR-RNA is a guide targeted to a specific past invader.
Precisely. These CAR-RNAs then associate with other Cas proteins, often a nuclease like Cas9 or a complex like Cascade, to form an interference complex. This complex is now primed. If the same virus, or one with a matching sequence, tries to infect the cell again,
The cRNA guides the Cas nucleus right to it.
exactly. The RNA base pairs with the complementary sequence on the invading nucleic acid, DNA or RNA, depending on the specific CRISPR system. This perfect match signals the Cas nucleus to cut and destroy the invader's genetic material.
Stopping the infection before it starts. Adaptive immunity in bacteria.
It's truly remarkable. A sophisticated defense system built on genetic memory and RNA-guided targeting.
So, wrapping this all up, what's the big picture takeaway from this deep dive into bacterial regulation?
I think the main theme is really the exquisite balance and efficiency of bacterial life. Survival depends entirely on these multiple overlapping layers of control. You've got proteins acting as switches and dimmers. You have RNA molecules acting as sensors and inhibitors. You have small molecule second messengers triggering global shifts.
All working together.
All working together, constantly sensing the environment and adjusting gene expression to conserve energy and respond effectively. It's not just one switch. It's this incredibly complex, integrated network, ensuring they only make what they need when they need it.
And thinking about that integration, what does it really mean? I mean, it strikes me that the same basic toolkits get used over and over again.
That's a great point.
We saw a two-component system controlling chemotaxis, tiny adjustments in movement. And you could have a similar TCS, maybe more complex, initiating something huge like sporulation, a massive developmental change that takes hours.
Yeah, the versatility is incredible. Evolution has clearly taken these fundamental regulatory modules, TCS, DNA binding motifs, second messengers, RNA structures, and repurposed and combined them in countless ways to control virtually every aspect of microbial life, from metabolism to movement to defense to social behavior.
It really underscores the power and flexibility of these molecular tools that underpin bacterial survival.
Absolutely. Foundational regulatory versatility that really defines microbial life.
A perfect place to leave our deep dive for today. Thank you so much for joining us as we explored this really intricate regulatory blueprint of bacterial life.
My pleasure.
We'll catch you next time on the deep dive.